see is when we look at this, there's going to be zones within the germinal center, a mantle, a light, and a dark zone. This is going to be due to who's collecting where. If you look at the germinal center, you're going to have the light zone, a dark zone, and this mantle zone. That's what I was just talking about. The different cells are going to contribute to different signals that the B cells will be exposed to. The mantle out here is going to be relatively resting cells that have just been activated and have migrated, or they are no longer receiving the stimulating signals because whatever it is they respond to is no longer in the body, so they are losing the signal to stay stimulated and they are now quieting down. The light and the dark zone is going to be dependent upon whether we're looking at centrocytes or if we're looking at centroblasts. The centrocytes are going to have the follicular dendritic cells stimulating them. Trying to get them to become, excretion and antibody secreting cells. And then we have the centroblasts. These are rapidly dividing cells. Here the artist has, shown them as rather large cells because they're getting ready to go through mitosis. So they're going to increase in size as a means of preparing for cytokinesis. Now, it's not a one-way thing. What we find is that as the cells go from the mantle to the light to the dark, they can cycle back and through light and dark, depending upon where they're at during the cell cycle, depending upon which signals they're being stimulated against, or stimulated by, CXCR5, CXCR4. These are going to be coming from neighboring cells, like the follicular dendritic cells. The T helper cells we see here will be stimulating the cells to aid them in their differentiation. When you look at it, you can see here that the cells, depending upon where they're at in the germinal center, tightly packed, lightly packed. So we see a T cell zone, a mantle, light zone because they're lightly packed, and then this dark zone where they're truly packed in. You can see here light where they stained it, light zone, dark zone, depending upon what they're looking at. Here at the dendritic cells, the follicular dendritic staining red, and down here are the centrosomes, the centroblasts staining green. Now, once the B cells hit the, germinal center, they're going to start seeing hypermutations going to be induced. The somatic hypermutation, where you can either get the antibody showing a greater or lesser affinity for its antigen, as well as the beginning setting the stage for class switching. This is going to be due to signaling coming from the helper T cells. The helper T cell is there, well then guess what? The signals are going to be there. It's going to tell the B cell to progress through the phases, to progress through and mature. The lack of the signals, well, not so much. The lack of signal is going to tell that B cell time to die, and it's going to become what's referred to as a tingible body, a dead mass that will then be phagocytized, removed from the lymph node by a macrophage. So when you look at it, we've seen this before. An antigen binds to the B cell receptor. Some of it's going to be taken up, processed by the B cell, presented on MHC class 2, which is going to interact with T cell receptor with the correct epitope presenting the T cell receptor. It's going to be a stable MHC TCR interaction. You're going to get some co-receptors interacting that aid in stabilization. Here they're showing usually it's a CD4. Then you're going to get CD40, CD40 ligand, as well as cytokine release. Incorrect or no antigen interacting with the B cell receptor, or there's no stable interaction with the T cell receptor, then you're going to be going on the left track here. Stable means continue signaling. Signaling comes through in different waves and different stages, is going to tell the B cell to become a plasma cell, and then over time to become a memory B cell. If there is no cross-linking, no interaction, as I was talking about, well, that B cell will not be stimulated to stay alive. These things have a short shelf life. They need to interact with the correct helper T cell. No interaction, apoptosis will be triggered. You get the tingible body, which will be removed by macrophages. But you get the stable interaction, B cell receptor, T cell receptor with an epitope. You get CD4, then you get the CD40 ligand, CD40, and the cytokines. Well, then you get the push. You get the push to begin what we had called in a previous chapter class switching. So a naive B cell, which is originally showing mu, IgM, will then be pushed to switch. Naive B cell will go through and slowly change. Go through the steps to, switch from, like I said, IgM to IgG epsilon at the heavy chain. Depending upon the signals, LPS, LPS IL4, LPS TGF beta, you'll see this switch go from a naive B cell, which is going to stay as an IgM. LPS will increase IgM production. LPS as well as cytokines will increase antibody production, but then also push to a switch. Switch from IgM to gamma epsilon, or strong push from IgM to gamma 2 or IgA, depending upon whether or not you have where the infection is originating. So we find that the cytokines are going to push class switch. Depending upon which cytokine the B cell will be bathed in, will push from M to the different G's, the E, and the A. And which cytokine that's going to be expressed by the T cell will be dependent upon, generally, by the source of where that antigen's coming from. Now, one of the things that we have figured out is that these immune complexes that are going to be presented to the B cells, that they can then go and try to find a T cell to work with them. They're not, as I said before, they're not going to be processed by follicular dendritic cells. No, follicular dendritic cells is just going to collect on its surface a bunch of antigens that have been opsonized, coated in antibodies. As they show here, it's on the surface. It does not get taken up. So it literally just goes cell to cell and say, do you recognize this? We find that the follicular dendritic cells stay in one region of the germinal center. They don't go around to all of the areas in the germinal center. Here they showed where they radio-labeled follicular dendritic cells, and they stayed up here at the mantle going into the light region, a light area. The antigens are bound to receptors on the surface, and that's where they stay. Here we see a receptor that recognizes the constant region of an antibody that is bound to some form of antigen, polysaccharide, LPS, whatever. C3b, part of the complement cascade, has its own receptor, CR1, complement receptor 1. So FCR, remember this is the fragment constant of the antibodies receptor. These follicular dendritic cells are going to be coded in these two types of receptors and many others. So that way things coat their surface, but they are not going to be taken up. They're not going to be processed. Finally, B cells that go through the different areas of the germinal center and get bathed in different signals, and they mature. They survive, they keep getting the signals, they survive long enough, they become mature cells. So what we see here is that there are some signals that will say no, PAX5 and BCL6. PAX5 and BCL6 tell the B cells no. Slow down. You do not need to do this. These are internal. BCL is one of the apoptosis activating signals inside of every cell in your body. Well, as the B cells are getting bathed in signals from the follicular dendritic cells, PAX5 BCL6 gets downregulated. It gets blocked off. So then you turn around and you see an upregulation of IRF4F, which is a transcription factor, which then goes and calls for increased activation or increased expression of certain genes, and BLIMP1 is one of those. So BLIMP1 is actually, it's kind of weird because this is a repressor. So it's going to take time for the repressor to kick in, for the repressor to get expressed. So while you're going through and you're activating the cells, you're getting them to be memory cells, you're getting them to go through class switching, and then at the tail end, IRF4F goes and activates BLIMP1 that shuts down class switching. So they go through, the cells will then go through, do the class switching, and then here comes BLIMP1 as a big shutoff. So now they're stuck. Whatever class they switch to is now the one that as a plasma cell they will be stuck at.